Welcome. My name is Dr. Catherine Klapperich. I'm the director of the Center for Future Technologies in Cancer Care here at Boston University. The title of this module is Access, Equity, and Molecular Diagnostics. I'm going to give a short talk that just goes over some of the technologies we've been working on in my laboratory for the last 10 years. All of these technologies have one thing in common, and that is that they are aimed at delivering low-cost diagnostics, molecular diagnostics, to patients and doctors in low-resource settings. I'd like to tell you about my experience attending an HPV, human papillomavirus, screening clinic in southern India this past summer. My colleague, Dr. Raj Dewar, took me to this clinic in order to teach me how difficult it is to perform molecular diagnostics in low-resource settings. Geeta, the woman here on my right, is a pathologist. She serves this low-income community and many other communities like it in southern India. Geeta is one of very few pathologists available to look at slides of cervical smears from these patients. What happened on this particular day was women showed up at a central location, were screened at first through questionnaires as to their risk for having HPV, the causative agent of cervical cancer, and then were given cervical smears or pap smears at the point of care. These pap smears were put on glass slides in the traditional way and quick prepped and stored. Geeta was not able to read them until several days or weeks later. In this time, the women left the camp and were unable to retrieve their results except for by cell phone. Raj and Geeta both are in dire need of a way to give patients the results of their HPV screen at the point of care. Many women do not know what their status is of this disease, and many women cannot be treated directly at the point of care unless they do know their status. So one of the projects that the center is working on, along with Dr. Dewar, is on a rapid point of care diagnostic for HPV that could be used at the point of care. So that seems like a pretty difficult problem. Doing a test that requires you to break open cells, get out DNA or RNA, amplify that DNA or RNA, and then determine whether the causative agent is present. And doing all of that without plugging anything into the wall or having large numbers of skilled practitioners on site. I'd like to argue that it's entirely possible to do these things. And furthermore, it's entirely possible to do these things with many of the technologies that we've already developed in the clinic and use routinely today in much, much more complicated manners. So before we move on to talking about the individual technologies, I want to tell you a story about my own family. So these are my daughters. I don't know which one is which, but I do know that they are about 5-day-old blastocysts in this picture. So you may be asking yourself, why do I have a picture of my daughters as 5-day-old blastocysts? The reason I have these pictures is that I carry a balanced translocation, which is a genetic defect that makes having children difficult. The only way to determine whether or not an embryo carries a balanced translocation and will thus be relatively normal, like I am, or carries instead an unbalanced translocation where some of the genetic material is missing or extra genetic material is present, is by performing an extremely precise surgery on those 5-day-old blastocysts, or even earlier on an embryo in vitro. So in this video, you'll see the pipetter come in from the right, touch the embryo, and pull out one single cell. This is done under a microscope in a very precise manner. Once this is done, the chromosomes are taken out of that one cell and labeled. In this particular case, the individual has a 5-7 balanced translocation, and you can see here in particular on the abnormal chromosome 5 the extra material from chromosome 7. And then you can see on the abnormal chromosome 7 the extra material from chromosome 5. This karyotype tells us that the cells in that particular embryo carry a balanced translocation. If patients with this balanced translocation are viable and normal, then this pregnancy would be allowed to go forward. If an embryo with an unbalanced translocation were present, that embryo would not be implanted in the mother. This is an amazing use of this technology. We're taking one cell to look at one karyotype and determine from that one cell of a very small number of cells whether or not a pregnancy will be viable. That's an incredible feat. However, the technologies have not been disseminated down to people who need them for other more mundane conditions. I want to discuss for a moment the latest incidence of Ebola hemorrhagic fever in Guinea and associated areas. In March of 2014, the Ministry of Health in Guinea notified the WHO of an evolving outbreak. It was only on March 23, 2014, 49 days after the first case was identified, that first case was diagnosed using PCR at a faraway hospital in France. This disease has a 59% mortality rate, which has a major impact on the surrounding communities. It spreads quickly, and perhaps the most troubling aspect of all is that healthcare workers are among those who contract and die of this disease before it can be stopped. The PCR used to detect this disease is relatively simple and well known, but we're unable to apply it in the setting where it's needed. This is in very stark contrast to the molecular methods that we're able to use, for instance, in vitro diagnostics at a large city hospital in Boston. I would argue that we should be able to take molecular diagnostics and put them into a platform that is as simple as another pregnancy-associated technology, the pregnancy test. Many of you will have seen tests like this before. These are strip tests that detect HCG protein in the urine of pregnant women. The good thing about this test is that the longer a woman is pregnant, the more of this protein is in her urine. So the test becomes more sensitive over time as the pregnancy progresses. There's a very high amount of this protein in the urine, so it's a very easy test to do and to do cheaply and to distribute widely. What I argue is that we can use some of the same techniques used in these devices and also some of the lessons we've learned from using these devices to make simple devices for molecular diagnoses, for diagnoses of diseases and conditions that require the detection and analysis of nucleic acid targets available to people worldwide. So what are the three major things that we need to do in a molecular diagnostic or a diagnostic to detect nucleic acids? We need to isolate the nucleic acids. Then, in most cases, we need to amplify the nucleic acids. We can either amplify the nucleic acids by amplifying the amount of DNA and RNA that are there in the sample through PCR or another associated enzymatic method, or we can amplify the signal that the presence of those nucleic acids generates. Finally, we need to detect that signal. Doing all of these three things are necessary in order to create a point-of-care diagnostic that will detect a molecular disease at the point of care. In order to create a molecular diagnostic that can detect a disease using DNA or RNA from a human sample, we need to follow three basic steps. The first one is isolation from the human sample. We look at urine, we can look at swabs, we can look at blood. We need to get the DNA or the RNA or other nucleic acid out of the human sample. Usually there's not a large amount of this material in order to make a diagnosis. So we either need to amplify the nucleic acids that are there or alternatively amplify the signal that those nucleic acids generate. Finally, we need to detect these nucleic acids. These, of course, again, are pregnancy tests, and you'll see in a pregnancy test you can read out the answer either by looking for your little red line, which is actually absent here, so this is the control line, or through a digital method, which gives you a readout, yes, you're pregnant, or no, you're not. We'd like to know that answer, yes or no, for a number of different molecular or nucleic acid-based diagnostics in the field. So first, let's start with the isolation step. There are a number of different ways to effectively isolate nucleic acids from complicated human samples. In our laboratory, we use a method based on the boom method. We take a sample and we mix it with a lysis buffer and pour it over a solid phase extraction column that generally consists of an activated polymer or a silica solid phase. In this diagram, the silica solid phase is represented by these little round nanoparticles. In the presence of the high salt buffer, the nucleic acids bind to the silica solid phase preferentially, and the rest of the molecules in the sample can then be washed away using an alcohol wash. Finally, when you're ready to do the downstream processing or amplification step of the nucleic acids, you can elute the nucleic acids in a low salt buffer or water into the downstream chip. We've done this in a number of different configurations. Usually the solid phase extraction column looks like a white column inside of a clear microfluidic chip that we make out of thermoplastics. We'll talk more about this in detail at the workshop. In a scanning electron microscope picture, you can see the solid phase polymer, and if you look more closely, you can see the second phase silica particles. These are made using light-directed chemistry in channels that are already assembled. We showed early on that we could get good DNA binding and elution from these columns. We then went on to demonstrate that we could use these solid phase extraction columns and relatively simple lysis chemistries in order to extract a number of different nucleic acids from a number of different infectious agents in some very challenging human samples. We've looked at nasowash in influenza A, we've looked at urine, blood, and stool, where we're looking for C. difficile, which is a gram-positive bacteria. And finally, we've looked at vaginal swabs for chlamydia and gonorrhea. Today I'll talk about a few examples of all of these in order to describe some of the simple technologies we've made that we hope will eventually enable point-of-care molecular diagnostics in low-resource settings. So the first study I'd like to talk to you about goes into amplification. We made a chip, in short, to look for the influenza A virus in nasopharyngeal washes and nasopharyngeal swabs. The chip we made does several things. In this picture, you can see that the sample goes into the inlet. We use a nasopharyngeal swab or wash sample. It then goes over the sample preparation column, which is just like the column that I showed in the last couple of slides, to grab and release the RNA from any flu viruses that might be inside of the sample. Finally, we elute those nucleic acids into a chamber where reverse transcription takes place. Once reverse transcription is completed and cDNA is copied from the sample, we then proceed with a PCR reaction. What's happening here is a continuous flow PCR reaction where the sample is going from a cold to a hot to a cool to a hot to a cool to a hot. And every cycle that the sample travels through the serpentine channel is a cycle of PCR. In this particular chip, it takes about 20 minutes to perform 30 cycles of PCR. At the outlet, we take the sample out and we look for the amplicon using capillary electrophoresis that would indicate infection with influenza A. The chip looks like this. It's made out of thermoplastic, it's clear, and the solid phase extraction column is sitting right up here in the chip. We used this chip to perform a clinical study in Boston during the flu seasons starting in 2008. Our study encompassed the flu pandemic. And so while we were in the middle of using these chips to detect whether or not our patients had influenza A in their nasopharyngeal wash or nasopharyngeal swabs, we were also being faced with the novel H1N1 virus in the community and the fact that the rapid immunoassays or the protein-based tests for influenza A and influenza B that were being used in the clinic at that time were not very sensitive. You'll see from this plot that in our data, we only had 50% sensitivity using the lateral flow strip tests, the protein-based test for influenza A and influenza B. The specificity was high, but the sensitivity was low. And when you hear Andy Phan's lecture, you can review what sensitivity and specificity mean before class. Our chip, however, the microfluidic assay, performed quite well. Remember, we're performing RT-PCR in the microfluidic platform, and we're comparing it to RT-PCR in a tube. Our sensitivity was 96%, and our specificity was 100%. This was an excellent result. However, you'll notice from the pictures that I showed of the chip earlier that this chip is actually quite complicated, even though the chip itself, this part of the chip here, is quite simple. It requires 2 heaters. The heaters need to be turned on at a particular time and turned up at a particular time in order to transition from the RT step to the PCR step. And the sample needs to be pumped through the chip using a rather large pump in order to get the appropriate flow. The sample then needs to be removed from the chip for capillary electrophoresis on another chip. Although this could be done in an integrated system eventually, it would still represent another complicated aspect of the instrumentation that would require sophisticated control mechanisms. So we asked ourselves, what can we do to take some of the things that we learned from making this chip and use them in order to use them in simpler ways in order to enable molecular diagnostics in global health. One of the first things we did was take the isolation step and take it out of that entire chip and say to ourselves, can we do the following? Can we do DNA and RNA extraction without any wall power? And the answer was yes. We were able to take a very simple configuration of syringes, there are 4 in this picture, but only 3 are needed to do a simple extraction, and pass our sample over a solid phase extraction column, just like the ones we made in the microfluidic chips earlier, but make them instead now in a pipette tip or some other small configuration that can be taken out of the device and sent to another location for downstream testing. We made this device. It looks complicated in the picture, but it does exactly what that little tiny chip did without having to plug anything into the wall, and that is we can extract nucleic acids from a sample introduced by a standard syringe into the device and extract the nucleic acids so that they end up resident on this solid phase extraction column, which is about the size of a pipette tip, which can be removed from the device and shipped around in a little plastic bag with some silica gel in order to keep it dry. This entire device is run without plugging it into the wall. The power comes from a bicycle pump that's attached to this valve here. We pressurize the chamber, and that provides enough pressure to push an entire 5 milliliter blood sample through the chip. We're now working with an industrial designer in order to make this a prettier system that could be more push button for people in the field. The next thing we wanted to do is show how we could apply this technology, and this is some very exciting data. So the technology here, you'll remember, is a portable nucleic acid extraction device. What's interesting about this data is this is HIV viral load testing using this portable device on human whole blood without plugging anything into the wall. So we take the human whole blood sample and we mix it in this device with lysis buffer. We extract it on this little tiny pipette tip that's attachable and detachable from the device. And we then extract that nucleic acid once it's clean from this pipette tip and measure it. We're able to show across 4 logs of HIV viral load that we have statistical difference between the logs, and that so we can detect viral load, and that we do as well as, or in some cases better than, a standard QIAGEN kit, which requires a number of different centrifuge steps and buffer changes. Perhaps the most exciting part of this technology is the following data. So not only can we store RNA from HIV viral load samples taken in the field without plugging anything into the wall, but we can also store that RNA First for a day at room temperature, which is 22 degrees C in our setting. Then for a day at 37 degrees C, and perhaps more exciting, for 37 degrees C for 1 week, and still maintain quantitative output. What that would allow us to do would be, instead of taking blood samples at a far-flung location and trying to maintain those blood samples in a cold chain before they could get to a centralized location for testing, we instead can extract those samples at the point of care and store them at the status of the technology right now for up to a week before we have to extract them and perform the downstream assay. This would allow people then to pick and choose what molecular assay they would like to do. It might also allow long-term storage of nucleic acid samples for surveillance activities. But of course, we would like to do amplification of the nucleic acids in the field, and how we go about that. It turns out there are a number of amplification techniques that can be performed without thermocycling, like the PCR in the influenza example at the beginning. One of those is helicase-dependent amplification. Helicase-dependent amplification is an amplification method that works a lot like PCR. You start with double-stranded DNA, and instead of using one enzyme, you use two. The helicase comes in and unwinds the double-stranded DNA. Once that happens, primers, just like in PCR, can anneal to the double-stranded DNA. The polymerase can then come in and march along and copy the DNA, creating two copies. If an elevated temperature is maintained, then the DNA copies itself just like in a PCR reaction. So we attempted to do DNA amplification without any wall power. How did we do this? Well, we took a microfluidic chip and we loaded it with DNA that we extracted using the system for nucleic acid preparation that I mentioned in the last few slides. And we put it in between two toe warmers from CVS. We enclose the toe warmers and the microfluidic chip inside of this calorimeter, like you would build in high school, and we controlled the amount of oxygen that would go in and out of the calorimeter by controlling how many holes we punched in the side of the calorimeter. What this allowed was the toe warmers to undergo their exothermic reaction at a relatively steady state and maintain the temperature at about 65 degrees Celsius, which is the temperature needed for the helicase and polymerase to do their reaction. Next I'll show you some data that we generated using this device. So this is amplification data showing DNA extracted from C. difficile infected stool using that portable no electricity nucleic acid extraction device that I talked about earlier, and then subsequent amplification using the cup-based device. And you can see from this gel that when we had positive C. difficile genomic DNA, we come up with the appropriate product. And when we have positive stool samples, we also come up with the positive product. And the negatives, water and water, are negative, and the negative stool samples are also negative. So we extracted a very difficult sample using a device that we didn't plug into the wall, and then we amplified those nucleic acids in a device that didn't require plugging anything into the wall. However, you'll remember there was also a detection step. And in order to do this detection step, or gels, electrophoresis gels in the laboratory, you know that we have to plug them into the wall. So the very last part that I'd like to talk about is how do we get signal out of these samples without plugging anything into the wall. This brings us back to the pregnancy tests that we talked about earlier. The great thing about pregnancy tests is you can read them out on a strip. The second great thing about these pregnancy tests is that you can read them out on the strip and everybody knows what they're looking for. They're looking for a little red line. They're looking for a little red line in the window. If we can translate amplified nucleic acids into a signal that looks like the output of a pregnancy test, I think we have a very good chance of getting our tests adopted in low resource settings. So let me remind you first here on this side how the pregnancy test works. So you have a clinical sample with the analyte of interest in it. As the sample flows along the lateral flow strip, which is just a piece of absorbent paper, the sample first comes into contact with an antibody that's attached to a colloidal gold particle. If the analyte is present in the sample, it'll bind to that antibody. Now you have an antibody that's bound first to your analyte and also to a gold nanoparticle, which when clustered on a paper strip becomes visible as the little red line that your eye sees when you're running a pregnancy test. Further downstream, you have the test line and you have the control line. The test line has antibodies against the analyte. So if the analyte is present, you'll have a nice sandwich assay, and gold nanoparticles will combine at this one location and provide a very red line and thus a positive test. In any test, whether or not the analyte is present, you have a control line against the gold particles, a control antibody against the gold particles. And those gold particles will line up along the control line, giving you a red line there, so that you know the test was run to completion and that it was run correctly. It turns out that you can also use nucleic acid targets, properly tagged, to achieve this same objective. So if, when you're doing your HDA reaction, you include a primer that has a biotin attached to it, and a probe that has a small molecule dye or another small molecule attached to it, you can create these same test lines that have antibodies to your analyte while you mix streptavidin-coated beads into the system, which will then cause them to aggregate on the different test lines to give you your red line output. This is an example of a positive test where both the control and the test are positive from a nucleic acid sample that was generated using an HDA with these specific primers. So now we have all three parts. We have isolation, amplification, and detection, and we have a way of doing all three of them without plugging anything into the wall, and a way of detecting them by eye. So the final example here is work done by my postdoc, Jackie Linus, looking at chlamydia. She built one of the HDA systems that I just described for chlamydia cells. And you can see, if you look at her paper strips, she has a control line and a chlamydia line for the amplified nucleic acids that she amplified using HDA. And she ended up with a lower limit of detection, somewhere between 10 and 100 chlamydia cells total in the sample. If you look at the amplification products from the HDA, you can also see on an electrophoresis gel the same kind of result. These bands down here are primer dimers. This is one of the dangers of HDA and why it's necessary to have a specific probe, like was described in the last slide, in order to make your test specific. Perhaps most exciting about this is that Jackie then proceeded to do isolation and amplification using a paper column instead of a plastic column like the ones we described earlier. She did not use the system for nucleic acid preparation that I showed you in earlier slides. She instead used extraction on a simple piece of paper, followed by helicase-dependent amplification on that same piece of paper, followed by elution of those reaction components onto the same papers that she designed to grab the amplified nucleic acid from chlamydia. You can see there's a decrease in the limit of detection when she does this method. However, she's able to detect 10 to the 3 chlamydia cells in a swab sample without plugging into the wall and only using chemical-based methods on paper substrates. Remember, these are not protein-based tests. These are molecular tests. So I would argue that combined, these technologies and technologies like them can lead us to very simple, readouts by eye. You're looking directly at the test strip that runs out on the bench to diagnose diseases in a molecular way. So I'm going to leave you with this following quote to give you some food for thought about how technologies that we've developed in the laboratory here at Boston University and at other places around the country can be adapted and used through creative engineering to bring point-of-care molecular diagnostics to people in the United States and around the world. Thank you.